Hello everyone, my name is Purnima Singh, a PhD candidate from the Department of Environmental Studies, University of Delhi, India. Today I'm excited to share a part of my PhD dissertation work with all of you. The title of my presentation is Caught in the Crossfire, Host Shifts or Evolutionary Traps in Invaded Ecosystems, which aims at looking at the ecological consequences of invasive plant species for the native herbivores. We all know that the invasive species are one of the top worldwide threats to the native biodiversity. The introduction of the non-native plants can have negative impacts by outcompeting the native flora, reducing plant diversity, and shifting species composition towards non-native dominated habitats. Consequently, such disruptions trickle down to affect the native herbivores. Hence, it is very important to study the invasive plant and insect interactions as they alter the native herbivore assemblages along with the altering the ecosystem structure and functions. And they also modify the habitats and the resources that are available to the native herbivores. There can be several outcomes of the interactions. A. The herbivores may adopt readily to the novel host and may benefit from them, which is termed as the host shift or a host range expansion. B. The native herbivores may rarely exploit the invasives without any impact. And C. A mismatch occurs where the native herbivores they accept the invasives but at a cost of reduced fitness, which is called as evolutionary traps. Here's a conceptual framework taken up from Sunny et al. 2015 to provide a context to my questions. We know through the extensive research in invasion biology that the invasive plants, after being naturalized in a novel area, release certain compounds that could be attractive or toxic to the native herbivores. The secondary compounds, if found attractive by the native herbivores, might be incorporated in their diet breath, leading to host shift or host range expansion. This consequently leads to an increase in the insect population. This is essentially shaped by the preference and performance dynamics between the interacting species. Given that the insect accept the new host and they perform well on the new host in terms of the number of eggs laid, the larval emergence rates, and the larval survival, and so on, the performance exhibits a strong support for the host shift. On the contrary, the individuals might be initially attracted to the plants, but their experience reduced performance or fitness of their next progeny. Schleifer termed that as evolutionary trap, which leads to the reduced progeny and hence a decline in the insect population. Moving on, as I introduce my study system, here is a humorous illustration about the state of lantana invasion in India. Lantana camara is a native to Central and South America. It was introduced to India in the 1800s for ornamental purposes. It is now profusely spread across forest margins, scrublands, and even protected areas. It forms dense thickets and outcompetes the native undergrowth. Hyptis suavolens is yet another invader from Central America, first introduced for its medicinal benefits. It continues to outcompete the native and is gregariously spread along roadsides and agricultural margins across the peninsular and the central India. Some of the key characteristics of the two plants are its aromaticity. They are both abundant in secondary metabolites and volatile organic compounds. The native herbivore under investigation is a species of the tree crickets known as Acanthus indicus. It is chosen mainly because of its association with invasives such as Hyptis and Lantana. It is widely distributed across the country and they are soniferous, that is, the males make acoustic calls to attract the females, and these acoustic signals are species specific. From a taxonomic standpoint, Acanthus belongs to the order Orthoptera and family Grillidae. Globally, studies have explored their calling activity, host selection, and in some cases adaptation to urban and altered habitats. However, a little is known about how this genus interacts with the invasive plants in the tropical ecosystems. This gap forms the basis of our inquiry. Moreover, the available literature on the preference and performance and host shift studies are mostly from the order Lepidoptera, Coleoptera, Diptera, Hymenoptera, and so on. Not a single study has come from this order. First, the objective was to delineate the calling pattern of 𝙊𝙤𝙢 indicus from native and invasive host plants in the field. We quantified both the spectral, that is the peak frequency, and temporal, which includes the syllable period, syllable duration, chirp period, and chirp duration of the calls recorded from the two invasives, that is Lantana camara and Hyptis suavolens, and one native, which is Justitia adatoda. The results suggested that the peak frequency varied among the two non-native plant species, while the difference in the temporal pattern between native and non-native host plants was observed only in the syllable period. The study also quantified the choice of the calling positions of the individuals on different host plants. The native 𝙊𝙤𝙢 indicus chose non-native Hyptis suavolens leaves extensively as a site to baffle. Baffling is an alternate reproductive tactic which is predominantly found in the male tree crickets, where they amplify their signals to attract females. Our second objective was to understand the host plant preference of 𝙊𝙤𝙢 indicus in field, common garden, and lab conditions. The study sites, it was conducted in Rajaji National Park, Uttarakhand, Ratapani Wildlife Sanctuary, and Satpura Tiger Reserve in Madhya Pradesh. This is a schematic representation of the methodology followed. The calling individuals were psychoacoustically located, their calls were recorded, and the individuals were captured from the wild. The individuals captured were subjected to different host plants and choice experiment setups and to one plant types in the no choice experiments. In the lab, an olfactometer wide tube experiment was taken up where the plant materials on each site were given as a stimulant for them to make a choice. The preference of the host plant for calling, mating, ovipositing, and feeding by 𝙊𝙤𝙢 indicus reveals an array of host plant use. The proportion of the same is shown in the bar graph here. It is interesting to see that the incorporation of the invasive species in their diet breadth and in relatively higher proportion. Similarly, the calling of the indicus from various host plant species is shown here in this graph. In the choice experiments conducted in the common garden, we saw that they were mostly located on Hyptis. If you look at the preference for the oviposition, we found out that the number of oviposition holes was maximum in the Hyptis in the choice experiment setups, and similarly a similar trend was found in the no choice experiments where the number of oviposition holes was highest on Hyptis plant. In the olfactometer choice trials, females made choices on Hyptis. However, an equal number of females chose to go on the empty arm as well. In the Lantana versus empty, there was no clear choice between the both arms, and the same proportion of males and females made no choice and stayed at the base of the arm. In Lantana versus Hyptis, females moved comparable number of choices to both Lantana and Hyptis. To summarize, our findings suggest that 𝙊𝙤𝙢 shows both behavioral and chemical attraction to the non-natives, Hyptis and Lantana. One plausible explanation could be the specific VOCs that are shaping their choice of host plant. We aim to identify these VOCs using GCMS. Furthermore, we plan to complement our preference data with performance-based experiments where we'll measure the survival, growth, and reproductive success of 𝙊𝙤𝙢 indicus on native versus invasive plants. The two combined would present a more comprehensive picture of the ecological consequences of plant invasions for the native herbivores. I extend my gratitude to UGC and the Habitat Trust for the funding. Thank you for your time and attention.